subscribe to the V7 Fancy Theater. Thank you. You'll never be disappointed indeed. And today I just want to take you through the fluid compartments of the body and some little information about the electrolytes within the body. I know it will be less, but it will be exhaustive. Okay? So the V7 Fancy Theater at gmail.com. You can contact us for more and better videos. Alright, so yeah, that one is very good. It's a very good channel. You don't forget to subscribe, share to other students to also enjoy. Subscribe for more and better videos. Like us. Click here for daily notifications. We always post our videos. Okay, so first and foremost, I want to teach you about the fluid compartments. The body has majorly water, and this water is distributed throughout the body in different compartments separated by membranes. So if you look at this is distribution of total body water in a normal person. In biology or in science, the normal person we shall consider 70 kilograms person. Okay, so if a guy has 70 kilograms, you will find that the total body water is about 42 liters. And these 42 liters, they are contributing 60% of the body weight. Okay, and out of this, you'll find that the intracellular fluid is having 28 liters. 28 liters out of the 42 are found in the intracellular fluid, are found within cells. And this is contributing 40% of the body mass and 67% of the total body water. So if an objective comes, any type question, you just know that 28, 42, these two values are the origin of these values here. Okay, and maybe at this 70 kilograms. Okay, so extracellular fluid, you'll find that the ECF is having 14 liters. So 28 and 14 contribute 42 liters. So it is having 14 liters, which is just 20% of the total body weight and 33% of the total body water. The extracellular fluid is, we have part which is called plasma. That is around 25% of the ECF. And plasma is about 3.5 liters. And yeah, that is 5 and 8 in percentage. Okay, interstitial fluid, transular fluid, and mesenchymal tissue fluid. These are contributing 75% of the ECF. That is about 10.5 liters, 15% of body weight, and 25% of total body water. So you'll find that we're going to talk about a lot more about these fluids. We're going to see how these values are obtained. These values are obtained through a couple of experiments and some mathematics. So you'll see that you will learn more about the transular fluid. Where is it found? Mesenchymal fluid. Where is it found? So let's continue. Just stick to this video. You'll enjoy. Alright. So total body water based on gender. Water is the principal and essential constituent of the human body, as I've said before, and the total body water is about 10% less in a normal young adult female than that in an average adult male. Okay, so if the male is having 60%, the female is having 50%. Okay, that is the average. Let's continue. And this is due to relatively greater amount of adipose tissue in females. So females are having a lot of fat deposited around the bums, around the breasts, bellies, okay, mons pubis. They have it a lot. So this is the reason as to why they have a 10% water less than an adult male. In both sexes, the value tends to decrease with age. So this value of water tends to decrease with the age. Alright, so it means you have to take more water when you grow older, okay? And the compartments, look at this. I want to take you through this before we continue. So if I have intracellular fluid, which will say they are 28 liters, call it 40% of total body weight. this water can get out of the cells, or the fluid can get out of the cells. Fluid gets out of the cells, it becomes extracellular fluid, and that is said it is just as you can see it here, extracellular fluid, which is 14 liters, contributing 20% of the body weight. And this fluid, we have fluid within the blood vessels. We have fluid which is interstitial fluid, which includes the transular fluid and mesenchymal fluid. Then we have fluid in the lymphatics. We have the lymphatics here. Now we have this fluid getting out of kidneys to be excreted. Fluid which is getting out of the GIT as feces. We have fluid within glands which is secreted and also absorbed. We have plasma, okay? We have fluid in the stomach which is absorbed and not secreted as gastric juice and water. So we have a lot of fluid changes, but the main point here is that fluid is just separated by membranes, just a single membrane. You can see this is a phospholipid bilayer. So only membranes are separating the compartments. All right. So let's continue. Extracellular fluid compartment constitutes about 10% of the body weight, and it includes one plasma. Plasma is the fluid portion of blood, call it, and it's the intravascular fluid, and comprises about 5% of the body weight. Okay, so this is around 25% of ECF is plasma. On an average, out of 5 liters of total blood volume, 3.5 liters is plasma. So the total blood volume usually is 5 liters, but the 3.5 liters are plasma, okay, the fluid portion of blood. The rest are soil cells. 2. Extracellular fluid includes the interstitial fluid, which includes also the lymph, and it constitutes the major portion, about 3/4 of the ECF. We'll say 25% is plasma. Then now 3/4 of ECF is the interstitial fluid, and the composition of ECF is the same as that of plasma, except that it has little protein. Thus, interstitial fluid is an ultrafiltrate of plasma. All right, ultrafiltrate, filtration under pressure. Okay. 3. We have transular fluid, which is contained in the secretions of secretory glands or secretory cells and cavities of the body. For example, transular fluid will be meaning saliva, sweat, cerebrospinal fluid, intraocular fluids, that is aqueous humor and vitreous humor, pericardial fluid, bile fluid present between the layers, pleura, peritoneum, and synovial membrane, lacrimal fluid and luminal fluids of the gut, thyroid, and cochlea. So all these fluids secreted by the membranes within into cavities, or by secretory cells, all that is transular fluid. A transular fluid volume is relatively small, just about 1.5% of the body weight. That is 15 milliliters per kilogram of body weight. That is 1 liter in a person of 70 kilograms. That it will be. So 1 liter constitutes the transular fluid. And 4, we have mesenchymal tissue fluid. Mesenchymal tissues are those, for example, the dense connective tissue, cartilage, and bones. These contain about 6% of the body water. Okay, and the interstitial fluid, transular fluid, and mesenchymal tissue fluid combinedly form 75% of the ECF. So you now have to note that the normal distribution of total body water in the fluid compartments is kept constant, and this is done by just two forces. We have osmotic pressure and also have hydrostatic pressure. So the measurement of body fluid volumes. Theoretically, it is possible to measure the volume of each fluid component by injecting a substance, call it an indicator, that will stay in only one compartment, provided the concentration of the substance in the body fluid and the amount removed by excretion and metabolism can be accurately measured. So the major point here is we need an indicator, okay? We need an indicator. We inject it in one, and it should stay in only one compartment. That we need to study its volume. Okay, and then we shall calculate that the volume of distribution is that the volume of distribution of that indicator is A1 minus A2 over total over the concentration. Okay, where the V is the volume of the fluid compartment, and A1 is the amount of indicator which we injected in the fluid. A2 is the amount of indicator which was removed by excretion and metabolism. Also cater for that. And the C is the concentration of the indicator within the fluid. This can be measured properly. Okay, so let's continue. For example, in case if 150 milligrams of sucrose, that is the A1, is injected into a man of 70 kilograms, okay, 10 milligrams of sucrose were excreted or metabolized, and the concentration of plasma sucrose, when we measured, we got 0.01 milligrams per milliliter. Then it will mean that the volume of distribution of sucrose is 150 amount injected minus 10 which was excreted over concentration we found, and you get 14,000 milliliters. Okay, so that's how we do it. So what are the prerequisites of accurate body fluid measurement? For it to be accurate, one, we need to make sure that these characteristics of the indicator are maintained, which characteristics include: one, it should be non-toxic, should not damage the body cells; two, it must mix evenly throughout the compartment being measured; and three, it should be relatively easy to measure its concentration, because if it is hard, then the C is hard to obtain. So let's continue. 4, it must not have no effect on its own distribution and it must have no effect of its own on the distribution of water and other substances in the body. 5, either it must be unchanged by the body during the mixing period, or the amount changed must be known. Okay, should not be excreted, should not be metabolized. But if it does so, or if it is excreted or metabolized, this amount metabolized should be taken care of, should be noted, should be measured. And this method of the measuring we are carrying out is what I didn't say at the start, but I call it indicator dilution method. This is a method we use, indicator dilution. Okay, so how do we measure the total water volume? We've seen water volume is around 42 liters. How is this measured? The marker will be one which evenly distributes in all compartments of the body fluid. And these markers will have deuterium oxide, D2O, we have tritium oxide, and also have aminopyrin. These three are capable of distributing into all the compartments. Okay, take note of this structure here. The volume of total body water can then be calculated from the values of concentration of the marker within plasma. Alright, how do we measure extracellular fluid volume? This can be measured by injecting those marker substances which cannot enter cells but can freely pass through the capillary membranes. They don't enter cells, but they freely pass through capillary membranes, okay? And such that thus these can distribute evenly into all the compartments of the ECF. Such substances will include, we have one, reactive substances like sodium, chloride, you have 36 and 38, bromide 82, sulfate, thiosulfate, and non-metabolizable saccharides. For example, inulin, which you usually use, mannitol, and sucrose. So these are capable of traversing the capillary membranes, but they do not enter cell. Okay, so the most accurate method of measuring the volume of ECF is by using inulin. The values of ECF are calculated from the values of the concentration of inulin in plasma since it makes an important component of the ECF. We say 3.5 liters of plasma is blood, of blood is plasma. Okay. 3. We have measurement of plasma volume. How is it measured? The plasma volume can be measured by injecting those markers. Plasma volume can be measured by injecting those substances which bind strongly to the proteins in plasma and either do not diffuse or diffuse only in minute quantities into the interstitium. And these substances will have radioactive iodine, that is 131, and we have the dye which you call Evans blue. These two are capable of doing it, okay? So Evans blue, take note of it. The plasma volume can also be calculated from the values of the red blood cells, which can be measured using radioactive isotopes of chromium. You get this point? Values of the red blood cells, which can be measured using radioactive isotopes of chromium. 4. How do you measure the intracellular fluid volume? The fluid within cells cannot be measured directly, since there is no substance which can be confined exclusively within cells after intravenous injection. Therefore, values of ICF are calculated from the values of total body water minus the fluid in the extracellular compartment. Okay, this is it. TBW minus ECF volumes. Okay, how do we measure the interstitial fluid volume? Like ICF, the volume of interstitial fluid also cannot be measured directly for the same reasons. Its values can be roughly calculated from the values of ECF and plasma volume, as interstitial volume is ECF minus that fluid within plasma. Okay? Note that the ECF volume or intracellular fluid volume ratio is higher. You get this point? ECF intracellular, ECF ICF fluid volume ratio is larger in infants and children as compared to the adults, but absolute volume of ECF in children is smaller than that in adults. Okay? And lastly, therefore, if there is dehydration, it will develop rapidly more frequently and severe in children than in adults because their ECF absolutely is lower. Let's continue. So now let's talk about the ions within the body. If we measure them in milliequivalents per liter of water, you'll find that sodium is high in the extracellular fluid. That is 142 compared to 14 within cells. Then potassium is the highest in the cells. That is 150 compared to 5.5 outside cells. Calcium is high outside. That is 5 milliequivalents per liter, whereas within cells it is less than 1 milliequivalent. Magnesium is highest to inside cells, at least outside. Anions, we have chloride. Chloride is too much outside cells, 103, and just 4. And 4 is found and within cells. The bicarbonate is highest outside cells, that is 28, compared to the 10 inside cells. The phosphate is 4, and much of it is found within cells, that is 75. Whereas the proteins is just 1 gram per deciliter outside cells, whereas it is 5 grams per deciliter within cells. So what are the functions of electrolytes? We know that electrolytes are the main salts in the body fluids for maintenance of acid-base balance. Okay? And 2, electrolytes maintain the proper osmolarity and volume of the body fluids. Alright. 3, the concentration of certain electrolytes determines their specific physiologic functions, e.g. the effects of calcium ions on the neuromuscular excitability. I hope you get this point very well. Okay? Their concentration determines specific physiology or specific physiologic functions. And it is important to note that essentially all of the body potassium is in the exchangeable pool. Only 65 to 70% of sodium is exchangeable. And almost all the calcium and magnesium are non-exchangeable. Okay? So potassium is exchangeable, can go in out. Only this is exchangeable in out of the cells. Okay, and only the exchangeable salts are osmotically active. So calcium and magnesium osmotically they're not very active, okay, because they are inexchangeable, non-exchangeable. Okay, don't you forget to like don't you forget to like our videos, subscribe for more and better videos, share to other students to also enjoy, share to other people. Enjoy the science we give at our channel. Subscribe for more and better videos. We have very good videos. Click this button for notifications. Okay.